Hello everyone, a very good evening to all of you. In this session, I will teach you a topic from general microbiology, that is, I will finish staining today. So, I will teach you the remaining portion of the staining techniques. So, we have already started staining in microbiology and we have studied the classification and most of the stains. So, this is the classification of staining in front of you. We have seen there are 4 types of stains: simple stain, negative stain, impregnation stain and differential stain. Till now, we have already seen what are simple stain. Simple stain are the stain which give same color to all the bacteria. What are negative stain? Negative stains are the stain which don't stain bacteria, which stain the background, so that in contrast of the dark black background, the bacteria stands out. The transparent bacteria stands out. Here you can see what is negative stain. It's very important. And the third type of the stains we have seen is the impregnation stain, in which the thin bacteria, on the thin bacteria which we do the layering to make them thick, so that they become visible. So impregnation is the layering basically. After that, the fourth type of the stain is the differential stain. Differential stains give different colors to different bacteria. So there are 3 types of differential stains. Out of the 3, we have already covered gram stain, acid-fast stain. In this session, I will be teaching you first Albert stain and then some special stains like bipolar staining and all. So in the gram stain and acid-fast stain, we have already covered in the last session. We have seen its principle, interpretation, diagrams, everything. Now it's time to start the Albert stain. What is Albert stain? The last stain, Albert stain. So Albert stain stains only one bacteria in the world. The name of that bacteria is Corynebacterium Diphtheriae. So it stains the diphtheria bacteria. It is only for one bacteria, not for others. So only one bacteria is Albert stain positive. That is the first thing you should learn, right? So that is the Albert stain. Now, what is the method? How do you do that? So first, take the smear. Take the smear of the bacterial suspension. On that smear, first you heat the smear. After that, put Albert 1 stain. Then without washing, put Albert 2. Put Albert 2 and without washing, just dry blot and see under microscope. Now you will ask me, ma'am, what is Albert 1? What is Albert 2? Can you tell the composition? So here is the composition. So this is Albert 1 stain. Don't learn the entire composition. Don't learn how many gram, milligram. No need to learn that. Just learn in Albert 1 there is toluidine blue and malachite green. So these are the main composition of the Albert 1 stain, apart from which glacial acetic acid, alcohol and water are there. So main things are toluidine blue and malachite green, that is Albert 1. In Albert 2 stain, there is mainly iodine and potassium iodide apart from water. So don't learn the milligram, ml and exact quantification. So that is about the Albert 1 and Albert 2. So heat the smear, put Albert 1, without washing put Albert 2, without washing just see in the microscope after dry blotting. So that is the method actually. Now in the microscopy what you will see? You will see like this. This bacteria is Corynebacterium diphtheriae. As I have told you, only one bacteria is Albert positive. So this bacteria is Corynebacterium diphtheriae bacteria, right? Now what is the color of the bacteria? In this, few important points are that the bacteria is green in color. I can see the green bacilli. The bacteria become green color, not red, not blue, not violet. No, it is green color. The bacteria is green color, right? Number 1. Number 2, the bacteria are angulated. I can see in the shape of the L and V. The bacteria are angulated. You can see this image or see this image. Most of the bacteria are angulated. Now the question will be, Viva question will be, why they are angulated? Why the bacteria are angulated? Actually, the bacteria, this is a bacteria, right? This is a bacteria. This bacteria is trying to divide by binary fission. So while doing binary fission, the 2 daughter cells are formed. This is daughter cell 1. This is daughter cell 2, but they are not separated completely. So incomplete separation during binary fission give rise to angular appearance. So angular appearance is due to incomplete separation of the daughter cells. You got my point? That's why they are either K, L or V shape, like angulated. So that is an important point. Number 1. Number 2 is the color. The color is green. You can see everywhere the green color. Number 3, let me draw a bacteria for you. Give me a minute. So can you see bacteria are like this? They are green color, number 1. They are angulated, number 2. Number 3, at their ends, at their poles, you can get purple color granules. The purple color granules are present at the end, at the pole. So let me talk about these granules, very, very important. What about these granules? What are these granules which are present at the ends? So you can see, since these granules are present at the poles, North Pole, South Pole, these are known as polar bodies or polar granules. Number 1. Since they are violet in color, they are known as metachromatic granules. Metachromatic means the color is violet or bluish. So number 2. Number 3, they were discovered by a scientist known as Babes. Beautiful name. Babes-Ernst. That's why these are known as Babes-Ernst granules. Number 3. And number 4, they were not discovered in this bacteria. This bacteria is Corynebacterium diphtheriae. But apart from Corynebacterium diphtheriae, it is present in some other bacteria also. The same granules I am talking about. So it was discovered in a bacteria known as Spirillum volutin. That's why known as Volutin granules. Volutin. So these are the 4 synonyms, the names given to these granules. What are the 4 names? Understand the meaning why they are given these 4 names. Right. Number 1. Number 2. Okay, you will ask me. Okay, ma'am, we got it. They are present at poles. That's why polar. They are blue in color, violet in color. That's why metachromatic. They were discovered by Babes-Ernst. That's why Babes-Ernst granules. And they were discovered in a bacteria. That is Volutin. That's why known as Volutin. But what are they? What are they? What does the composition? They are made up of phosphate. Actually, the phosphate in the bacteria is accumulated in these granules. Now, you will ask me, ma'am, why the phosphate is accumulated? When the bacteria divide now, continuously divide, phosphate is utilized in the nucleus formation. For the formation of the nucleus, for duplication, replication of the nucleus, doubling the nucleus it is required. But during starvation, when there is starvation, bacteria don't divide. When the bacteria is not dividing, where the phosphate will go? Phosphate is not utilized for forming the nucleus because duplication of the nucleus is not taking place. So phosphate will accumulate. So these granules will be visible in bacteria only at the time of starvation when bacteria is not dividing. So that is the all concepts about it. Everything is written in the notes. See, have a look. Have a look. So what are these granules? Tell me. What are, before that, one more important thing. One more. As I have told you here, the bacteria is in angular fashion. Can you see the bacteria is in angulated fashion. Because of this angulated fashion, it is looking like a Chinese language. Do you know Chinese language? No, at least I don't know the Chinese language. It is looking like something Chinese language. That's why known as Chinese letter arrangement. This arrangement known as number 1. It is also looking like cuneiform. You know, it is looking like cuneiform. That's why cuneiform arrangement. So Chinese letter or cuneiform arrangement is a very important topic. So what is Chinese letter arrangement? It can be a 2-mark, 1-mark question. What is cuneiform arrangement? So number 1, this arrangement is seen in Corynebacterium diphtheriae. This arrangement is seen in Albert stain, Due to incomplete separation of the bacteria, they are angulated. So Corynebacterium diphtheriae appears green color along with angulation, and at the ends bluish granules are present. And this arrangement is Chinese letter arrangement or cuneiform arrangement. The granules are, that is about the granules. So what are the different names given to the granules? The granules are known as Volutin granules. That is the name of the bacteria in which they were first time discovered. Babes-Ernst granules. That is the name of the scientist who has discovered it. Polar granules because they are present at the poles. And metachromatic granules because they are bluish in color, violet in color. So that is the names given to them. The meaning of all these things are same. Actually, they are deep blue in color and they are phosphate. They are the stored phosphate. Why the phosphate is stored? During starvation, the bacteria is not dividing. So nucleic acid synthesis is not taking place. So phosphate is not utilized. That's why it will accumulate. So basically these granules are the intracellular phosphate reserve when nucleic acid synthesis is not taking place. So it is a very important question that can be a small 1-line question. Now can you tell me 5 bacteria in which these granules are present? So there are 5 granules. You know one of them, Corynebacterium diphtheriae. They are present in Corynebacterium diphtheriae. I have shown you the image. But apart from Corynebacterium diphtheriae, it is present in 4 other bacteria also. So I am having a mnemonic again for you. The mnemonic is MSC Garden in Agra. In Agra, there is a garden. The name of the garden is MSC. You can learn like that. Actually, it is a garden. Yes or no, I don't know. Right? I make a mnemonic. So M stands for Mycobacterium, tuberculosis, leprae. S stands for Spirillum volutans. This is the first bacteria in which it is discovered. Right? You know that. That's why these are known as Volutin granules. Right? C is Corynebacterium diphtheriae. You know that. Right? Garden is Gardnerella vaginalis. And Agra is Agrobacterium tumefaciens. So these are the 5 bacteria in which these granules are present. Can you tell me the name of the 5 stains with which we can stain these granules? You know one of them. One of them is Albert, with which the granules are blue in color, right? We have seen the diagram. But apart from Albert, 3 more stains are there. Again a mnemonic. The mnemonic is PLAN. P stands for Ponders, L stands for Loeffler methylene blue, A stands for Albert, and N stands for Nizar. So this is all about the Albert stain. Let me summarize Albert stain. Before leaving everything, you should always make a habit to summarize. So what is Albert stain? Can you tell me everything about the Albert stain? What you have learned? So you have learned that Albert stain is only for one bacteria, that is Corynebacterium diphtheriae. What is the procedure? You use two stain. Albert 1, you know it contains toluidine blue and malachite green. And you use Albert 2, which contains iodine and potassium iodide. So that is the procedure in which you stain the smear with Albert 1 followed by Albert 2 and you see in microscope. Can you describe the microscopy? How does it look like? Can you describe the microscopy of Albert stain? Yes. Number 1, bacteria looks green in color. Number 2, bacteria are angulated like L, V, like that. The reason is incomplete separation of the bacilli. Number 3, it looks like a Chinese language. That's why known as Chinese or cuneiform arrangement. Number 4, at the ends of the bacteria, certain granules are present, which are blue color, violet color. The names of the granules you already know. So that is the microscopy. So only one bacteria, Corynebacterium diphtheriae, and this is the microscopy, this is the procedure. So that's all about Albert stain. But as I have told you, it contains granules. So ये granules पे short note अलग से. You can get a question on these granules separately. Granules को पकड़ लो. So what are these granules? Can you describe these granules? So describe 4 things. Tell me the other names given to these granules. Tell me the composition and reason why the granules are formed. Tell me the composition. Tell me the reason. Tell me all the bacteria in which these granules are present and tell me the stains by which I can stain these granules. Now, in your exam, you can get a separate question on Albert stain and you can get a separate question on these granules. Can you tell me the answers? So can you tell me the different names given to these granules? Yes, they are known as polar granules. These are known as Babes-Ernst granules. These are known as metachromatic granules and these are known as Volutin granules. You know the reason. The composition is the phosphate. They are stored phosphate. The reason, during starvation, when the cell is not dividing, the nucleic acid synthesis is not taking place, so phosphate get reserved. It get accumulated. Name the bacteria, the mnemonic. The mnemonic is MSC Garden Agra. Can you try to see the full form? That is the mnemonic. Can you tell me the stains? 4 stains. The mnemonic is plan. You know the plan. Ponder, Loeffler, Albert, Nazar. Right. Now you get these questions in your university exam also. Small questions. Either very short or short. Or you get MCQs on that. Now in your exam, you can get a question on Albert stain. Write all these. If you get a question on polychromatic granules, polar granules, write all these. Or you can mix the two. Right. So that is all about Albert staining. So we have finished the 3 differential stain, Gram stain, acid fast stain, Albert stain. So we have finished all about staining. So simple stains are done, negative done, impregnation done, the 3 type of differential also done. Gram stain, acid fast stain and Albert stain. Everything is done. Now the next topic is the bipolar staining. What is bipolar staining? Does anyone know what is bipolar staining? It is also known as safety pin appearance. Have you seen safety pin at your home? You all have seen it, I guess. This is a safety pin used for multi-purpose. So it is safety pin. This is known as safety pin. The bacteria are looking like safety pin. How come? How come the bacteria look like safety pin? Can you see? These are the bacteria. Let me draw the bacteria. The bacteria. Now inside these bacteria, the stain. Where is the stain present? The stain is present at the 2 poles, not throughout uniformly. The stain. Now this stain can be any stain. It can be Gram stain, acid fast stain, Albert stain, any stain, simple stain. Alright, the stain is accumulated at the poles. So it is looking like the safety pin. It is empty from the center and the stain is accumulated at the poles. See the diagram. Can you appreciate? Can you appreciate? These are the bacteria. Where is the stain? The stain is at the poles. That's why this arrangement is known as safety pin arrangement. Safety pin arrangement or bipolar. You got the meaning of the bipolar staining. The stain is present at the 2 poles, north pole, south pole, not throughout. So bipolar staining also known as safety pin arrangement. It can be any stain. Now you will ask me, ma'am, name the bacteria which shows this arrangement. Can you name the bacteria? This is the list. Now it will be difficult to learn the list. Again a mnemonic. I am having many mnemonics for all of you. So the mnemonic is in front of you. I have made this mnemonic for you. Right. So safety pin can produce very high yield. This is the mnemonic. If you have a better mnemonic than this, easy to learn or more interesting, more catchy, you can please drop that in comment. I will share with other students in my next lecture, right? So please try to make mnemonics as much as possible. It will be easy for you to learn with the help of mnemonics. So can produce very high yield. C stands for Campylobacter granulomatis, right? P stands for Pseudomyeli. It is Pseudomyeli, Pseudomonas pseudomyeli. And V stands for Vibrio parahemolyticus. It is not Vibrio cholerae. It is not Vibrio cholerae, mind my words. It is Vibrio parahemolyticus. H stands for Haemophilus. It is not Haemophilus influenzae. It is Haemophilus ducrei, right? And Y stands for Yersinia. So can you help me with the mnemonic? Safety pin can produce very high yield. So this is my mnemonic. Safety pin can produce very high yield. Can you help me? So C stands for Campylobacter granulomatosis. P stands for pseudomyeli, V stands for Vibrio, but not cholerae, Vibrio parahemolyticus, right? Because in your options you will get cholerae also. S stands for Haemophilus, but not influenzae, it's ducrei, right? And Y for Yersinia, right? So that's it. So this ends with staining. So all about staining in microbiology, it is a very important topic for your theory exams, for your MCQs, for your competitive exams. You can't miss stains in microbiology, and without learning stains, you can't move ahead in microbiology with other bacteriology, right? Okay, so that forms the basics. I guess it's crystal clear to you. I request you to all watch all my 3 episodes on staining if you have missed any of them. So if you have attended my lecture attentively, can you answer 2 questions to me? Write down the answer in the comment box. Can you write the answers in the comment box? Question number 1. Question number 1. I want everyone to write the answer in the comment box. Bacteria with safety pin appearance. Can you tell me the name of the bacteria? Come on, with safety pin arrangement. So just now I taught you the mnemonic. Can produce very high yield. Apply this mnemonic. What is coming among those in this mnemonic? Is it H. influenzae? Is it Vibrio parahemolyticus? Is it Vibrio vulnificus? Is it Salmonella paratyphi? What among these is coming in this mnemonic? Apply it. Try it out. So this is question number 1. Please write down the specific answer of question number 1. This is question number 2 for all of you, right? Albert stain is used for. Tell me only one bacteria in the world which shows Albert staining. Only one bacteria in this world shows Albert staining. I told you. So is it Staphylococcus is the answer? Is it Corynebacterium Diphtheriae? Is it Clostridium perfringens or Clostridium tritanii? What is your answer? Can you write it in the chat box, comment box actually? So yes. So that is all about for today's session. You are free to ask your doubt along with your name if you wish to tell me your name, college name, university and exam dates, right? So my contact number is in front of you. You can connect me on WhatsApp or Telegram. It's 9833038948. Don't hesitate to contact, right? If you have any study related doubt, you can contact me, right? So before ending, as usual, I would like to give you a small motivational quote. To change your life, you should change your priorities. You want to change your life, right? You want to become a great practitioner one day, so that you want an honor of being a doctor. You want to become a doctor. You don't want to add simply Mr., Miss or Mrs. before your name. You want to add doctor as a prefix before your name. So you want to change your life. You want to change the life of the others. You want to save the life of the others. For that, you have to change your priorities. You have to change your priorities. Your priority right now should be concentrate on the studies. Do smart work and concentrate on the studies. Give your maximum output in studies. Whatever best you can give, you should give. You should get good marks, distinctions, gold medals in your university exam, and you should get a single digit rank in your competitive exams, so that you can do your MD or MS in your dream branch. By the way, what is your dream branch? Can you comment me? What is your dream branch? There are 19 subjects, 19 branches available. What do you want to become? You want to become a dermatologist or a radiologist or a pathologist or a gynecologist or an orthopedician or a surgeon or a medicine practitioner. What do you want to become? So can you tell me? So I will be obliged if you tell me. I will be happy if you tell me. So you want to do your MD MS from one of your dream branch. You should be clear what you want to become. ये नहीं कि जो मिल जाएगा मैम चलेगा. चलेगा नहीं होना चाहिए life में. Don't compromise in life. I have never compromised in my life. At least for the branch which I have opted, right? I'm a pathologist and I'm proud to be a pathologist. I'm an oncopathologist. I do reporting of the cancer specimens. I'm a histopathologist specifically. So I have done my MD from one of the apex institute, not only of India but of Asia. I have done my MD from Tata Memorial Hospital, Mumbai. So I'm proud to tell you that. And by choice, I have taken that. By choice, that is my choice. I have given multiple attempts in my competitive exam. At my time, it was not NEET PG, it was not next. At my time, it was All India PG. So in my first attempt, when I have given that along with the internship, I got OBG, MS OBG. But I worked for a few months. I feel that it is not my cup of tea. So I changed my branch and I given the exam again. After that, I took anesthesia also. I worked in anesthesia also for a few months. But in the same year, during the second counseling, again I found out, no, it is not my cup of tea. Then I took pathology, and one of the best institute I took pathology, I found it is. So decide what do you want. As I don't do the mistake which I have done. I was very indecisive what I want. I want gynae or I want pathology, I want radiology. What is my choice? So you should be clear in your intentions what you want to become, because life of every branch is very different. It is not कि सबकी life एक जैसी होती है. The money is different, the life is different, emergencies are different, what you are living is different, right? So decide what you want, and whatever you want, do it from one of the best institute, one of the best. तो मजा है, right? For that, change your priorities in life, at least for the next few years, right? So okay, that's all about today. If you like the lecture, don't forget to click on the like button, share with all the medicos these links. You can share the link of my YouTube channel with all the medicos throughout the globe, you know, in your contact. And don't forget to subscribe and click on the bell icon before leaving, so that you will be notified for the next session. Thank you. See you. Bye bye. Study hard. All the best.